Hello everyone, Kent Bressler here. I want to welcome you to Kent's Kidney Stories. During our time together over these podcasts, I'd like to discuss kidney disease. I'd like to tell you about my journey as a transplant patient, but also talk about dialysis, kidney donation, and just about anything else that might be of interest. Kent's Kidney Stories, a podcast endorsed and sponsored by KidneySolutions.org. Hello, everyone. This is Kent Bressler, and we're back again to do some podcasting. Got a great guest today. We're going to talk about xenotransplantation, but before we do that, before we even get started, let's go to our Father and talk to him. Dear Heavenly Father, we look to you for guidance, for peace and love, knowing that it'll always be there. Keep us safe in your arms as we continue our separate journeys. Bless those in need, and may they come to know you in their time of need. We ask this in your holy name. Amen. I really do appreciate the listeners out there and want to tell you there are a lot of things going on in the kidney world. And as you well know, although COVID is on the decline, let's stay diligent and let's watch out, take care of ourselves and make sure we're vaccinated and that we're paying attention to crowds and at least distance and just take protection for you and your families. Today, what we're going to talk about, and I really have a great guest, and I've worked with him a little bit. I'm going to get to work with him a little bit more. So we're going to start today by introducing our guest, Dr. Michael Curtis, the president and the CEO of a company with a great track record in xenotransplantation, and that's eGenesis. And in order for me to get started here this morning, I wanted to read their vision statement, which I extracted from their website, eGenesis.com. At eGenesis, we believe that xenotransplantation has the potential to transform the treatment of organ failure by eliminating waitlist mortality. We believe that our gene editing platform will eventually reduce or eliminate the burden of post-transplant immunosuppression. We believe that one day, failing organs will be replaced with the same predictability and success seen with hip and knee replacements today. And as we well know, 14 people a day die waiting on a transplant. That's a large number. Large number of those are kidney patients. All right. And we also know that hip and knee replacements are common day. It's an everyday occurrence now. And we hope that at some point in time, just like eGenesis does, that xenotransplantation could supplant that. Okay, so without further ado, I hate that word, but I did it, is Dr. Mike Curtis. Mike, great to see you this morning.
Kent, great to see you. Thanks for having me on. I appreciate it. It's great to be here.
Well, this has been something that's been very interesting to me. I've had kidney disease all my life. I started out at a young age, and although I didn't know about it, I progressed fairly quickly to the choice between transplantation, dialysis, or death. And I believe me, when we have alternatives in this field, in the field of transplantation and kidney disease, it's great to have eGenesis as one of the many programs that are really working hard to help the patients. It is a patient-centered objective. Is that correct?
Yeah, no question. I mean, we leverage some of the breakthroughs in science. So a lot of the story is around CRISPR and how we edit donors and all that. But at the end of the day, the point is really to help patients, waiting for a kidney transplant and get patients off dialysis.
That's I think that's about as close to a cure as you're going to come. I know that a lot of things, diseases in the kidney world especially, And I know there'll be issues with heart transplants and other things like that. But in the kidney world, there's not many options. You have very few. In the last 5 years since I've been in it, I've seen a tremendous increase in the amount of options for patients. This is just another option, and it's not to be taken lightly. It's in its serious form right now in its production and evolution. So tell us a little bit about your background, first of all, and then let's talk about eGenesis and what xenotransplantation is really working at.
Sure, no, happy to. Yeah, so I'm lucky to have been in the biotech industry now for just about 40 years. And maybe and most of that time was spent in translating new ideas for drugs into clinical trials and a couple approvals. I was lucky enough to be involved in the early days of some of the immunosuppression agents that are currently used in transplant. And about 2.5 years ago, the team at eGenesis came to me and said, hey, we have this idea for xenotransplantation. We think we're ready or close to ready to start the first clinical trials. Would you like to join? Again, my experience is all taking the first idea and getting it into those trials and trying to get the product approved. And to be candid, I was a little skeptical, right? Because the idea of using porcine donors as organ donors for patients seems very science fiction. And I know we always have this mantra in xeno. It's sort of like xenotransplantation is the future, and it always will be. How close are we really to helping patients? And then the eGenesis team shared with me some incredibly compelling data where they had taken a porcine kidney and put it into a monkey, and the kidney functioned for just over 300 days. And so I said, well, all right, that's pretty good. That's a pretty good start. And so to me, that was compelling enough to join the company and really step in and try to help, make this vision of xenotransplantation a reality. As we sit here today, and we can talk a bit about how we do this, but as we sit here today, just actually this past Wednesday, we had a monkey that's carrying a porcine kidney that has survived for 700 days post-transplant.
Really?
Right? And yeah, so our goal in using the primate as the model was really to show, compatibility and long-term graft function. And we believe, we now have the data that says there's a reasonable chance that this could work clinically and will work for patients, right? And I think one of the questions to patients is, what kind of durability in a transplant do you need to see to make it worthwhile? And I think it's very individual, the answer to that question, sure. Right? But having now some recipients approaching the 2-year mark, we feel like, okay, this seems real, and we think it's time to go to the clinic. This past July, we had what's called a pre-IND meeting with the FDA to talk about what the FDA needs to see to do the first clinical trial. We talked about how do you control the donor, right? So the porcine donor, we want to make sure we control what we call adventitious agents. So we want to make sure there are no diseases coming from the donor itself. So we need to make very clean donors. We also talked to them about what kind of primate data do they need to see to allow us to do the first trial, right? And so, of course, 12 months of graft function in the monkey is what they'd like to see. How many of those would they like to see? It's somewhere between 4 and 6 studies. And that data set, we feel would be enough to support the first in patients. And then the last part was what patients, who on the wait list could we put into the phase 1 safely, right? And we had proposed patients 55 to 65 on the wait list because when we look at the data, and I'm sure you guys are really familiar with this data, the likelihood of those patients being transplanted is low, right? Yeah. And so there's a huge unmet need there. The agency asked us to add a couple of things to try to get patients that are even in higher need. So one is patients that had failed allotransplant. So they had already had a transplant. That transplant failed. They found themselves back on the waiting list, back on dialysis. They'd like us to include those patients in the trial. And for us, and our CMO Ellie Katz, who's a liver transplant surgeon, who's been with us now about 6 months, one of his biggest concerns and our biggest concern is getting patients into the first trial that are healthy enough, right. For success in transplant. Because we know, unfortunately, you spend too much time on dialysis, transplants become more difficult. right? And so it's that balance of unmet need, but healthy enough to really test the technology. Because our biggest fear is if we go into patients that are too sick, does that override the ability to see how well the transplants perform?
Exactly. That's going to be the biggest. I sat through that entire presentation that eGenesis gave to the FDA.
Yeah.
And I mean, I was amazed at the camaraderie there was between the groups and how much information was given to the FDA. And Dr. Katz really represented, I think, the group very well when he said, we have to be very circumstantial here. We need to know what the right person's going to be. We understand that. And so you've been able to work with the FDA since then and get some clarification?
Yeah, so we know what the FDA wants, and I think there's still a little bit of debate to be had about whether the criteria they're looking for is what the patient population we want to study. And so, I think one of the challenges in this space is there's a lot of unknowns, right? Sure. We don't exactly know what's going to happen. The FDA doesn't know what's going to happen. They don't have a crystal ball, right? And so we need to work together to get to a place of comfort. And this is, I think, where the patient voice is really important, because we, in the absence of hearing from patients, we sort of make it up, right? We look at the data and we propose something. And in my experience, if we don't involve the patient in the thinking, we could show up with a trial that nobody wants to join, right?
Sure.
Like you've now created an unenrollable trial. So it's a balance really of the three, interested folks. It's us, it's the FDA, and it's patients, right? And we got to make sure that we address the concerns of everyone to run that first trial. The good news, I think, to your point about the camaraderie and the discussion is I believe, and I think the FDA has shown this over the past year, that we're all aligned on the unmet need and the need to do something.
Oh, yes.
We're also aligned on the potential. And I think the challenge just is how do you make the first steps? We were incredibly excited about the work that was done at the University of Maryland with the first ever porcine heart transplant into a patient, because it showed a couple of things. One, the technology is advanced enough to do that kind of a study. And two, the FDA agreed with the compassionate use approach. We have thought about compassionate use in the kidney transplant. The challenge in kidney transplant, as you guys know better than us, is dialysis is available, right? And so making a compassionate use argument, and compassionate use really involves, how close patients are to death. Often they're not, right? It's not, we don't have to do something this week, right? We have time, which works against a compassionate use argument. A patient like Mr. Bennett, who received the first porcine heart, he was, on ECMO for a while, he was approaching death. And the last chance for him was this porcine heart transplant.
Right.
In the kidney world, it's a little hard to make that argument, but as we talked about earlier, there's a lot of patients waiting, right?
Exactly.
So
over 100,000.
Yeah. So with the right safety data set, I think we can find, and we've talked to patients that would be, when we talk about the potential for a trial, are interested in participating. And for us, patient safety is paramount, right? Because you can spend time on dialysis, right? We want to make sure that if the transplant doesn't work for some reason, that patients can minimally go back on dialysis. And we've also had initial discussions with UNOS about having a xenotransplantation doesn't disqualify a patient from getting an allo down the road, right?
Right.
So we want to make sure,
yeah.
So we want to make sure that both of those things are taken into account when we think about the first trial.
One of the things that really amazes me about the process is how quickly it started to move. It's not quiet. It's actually starting to beat some drums. There are a lot of things going on out there in the world. Kind of tell us a little bit about how eGenesis looks at this platform, if you will. Looks at the way, I think it's called the eGen platform.
Yeah. Yep. So what's really happened in the past, to your point about like this momentum that the field has, I think part of that momentum has come from scientifically our ability to edit the porcine genome, right? So if you go way back into the early 90s, we didn't really have the tools available to improve the compatibility between porcine donors and human recipients. So like a good outcome in a monkey kidney transplant was 30 days, right? You could get kidney function for 30 days with pretty intense immunosuppression. And then in the mid 90s, right, the world discovered retroviruses in the pig genome, right? These retroviruses are similar, right, to HIV, right? And that's HIV started out as a retrovirus in primates and crossed over into people. And so when those were discovered, no one really knew what to do about them, right? Because the risk seems low. We've been living with pigs for millennia.
Oh yeah.
And we haven't had a retroviral transmission from a pig to a human, but we've also never taken a pig organ, porcine organ, and put it into a person,
Right.
and then put that person on immunosuppression. And so what would happen? And I think the worst thing would be if that patient got a retrovirus, and then that retrovirus crossed over into the general population, and now we've just created a big problem. COVID is not a retrovirus, but it is a virus that likely came from an animal, right, and crossed over into people, and then we have a global pandemic.
Yeah,
so I think it happens. So yeah. we don't know the risk, and so at the time in the mid 90s, the field sort of slowed down, and several countries put a moratorium on any work in xenotransplantation because they didn't want the risk of a potential what we call zoonotic event or crossover. And then in 2010, the discovery of CRISPR-Cas9 happens. And what CRISPR-Cas9 allows us to do is allows us to go into the genome and make specific edits to different what we call nucleotides, so to the DNA. We can edit the DNA of the porcine donor. And what George Church and the team at Harvard did was they took the porcine donor and they used CRISPR-Cas9 to inactivate the retroviruses in the porcine genome. So reducing the risk of this zoonotic transmission. Now, what was really amazing with this scientific advancement was in a porcine genome, you might have anywhere from 50 to 70 different copies of this virus. And until the discovery of CRISPR, there was no way you could hit all 70. And what George and the team showed is they hit all 70. So now you have a way that you can edit the porcine genome to reduce the risk of zoonotic transmission. So this retrovirus is now inactivated and can't transmit into people.
It turns off the key. The key turns off the genome.
It turns off the ability of the virus to replicate, right? So we basically make a mutation in a gene in the virus, and now the virus can't replicate anymore. And so we inherited or licensed that technology from George and his team, and that was the forming of eGenesis, was the ability to produce donors, porcine donors, that have the viruses inactivated. In addition, at the same time, right, the field was trying to figure out what are the other types of modifications that we need to make to the porcine genome to improve compatibility and promote long-term graft survival. And so in addition to the retroviral edits, right, the inactivation of the viruses, we take genes out of the pig genome, right? And so what you recently saw Bob Montgomery at NYU, right, do some stuff in what we call decedent recipients, Jamie Locke at UAB. They were focused on reducing acute rejection. So if you take a unedited porcine organ and put it into a monkey, it will be rejected in a couple of hours to a day. It'll just turn black and will stop working. What the field found in the 90s is there's several what we call carbohydrate antigens, so sugar molecules in the pig donor that are recognized by the human immune system. When these get recognized, there's an immune response and the organ gets destroyed. So to avoid that, the field now makes what we call the triple knockout. So there's 3 genes in the porcine genome that we inactivate. We use CRISPR to do that. And basically make inactive, and then we improve the carbohydrate compatibility or the sugar compatibility between the porcine donor and human recipients, and now all that acute rejection goes away. And that's what Bob and Jamie and Mohammed and the team at Maryland showed, was in all recipients in the past year, human recipients, there was no acute rejection, right? So all those organs survived for at least a day or more, which was a big validation because nobody knew that everyone assumed they would work, right? But you really don't know they're going to work until you get into people. And then the third group of edits that we do to the genome, and this really constitutes the whole eGen platform, is we then take human genes and we add them to the porcine genome. And we add genes that can control rejection, right? So when you think about organ compatibility and supporting long-term graft survival, there's things that we want to do to reduce the inflammatory response to the porcine organ. And to do that, we add human genes that regulate something we call complement, right? So this is a really powerful rejection mechanism that if you don't regulate it, the organ will get rejected.
Yeah, almost immediately, right?
Almost immediately, exactly. And then we also add genes that improve coagulation compatibility, right? So we want to make sure we don't have clots, in the organ. And then we regulate what we call innate immunity, so the rapid response to the organ, and then adaptive immunity, so immunity that comes in over time, right? So why do organs eventually get rejected? Because these slower processes for rejection take more time to happen. So we add genes to the porcine genome across all 4 different mechanisms of compatibility and rejection. So taken together, our porcine donors will carry upwards of 80 edits to address retroviral risk, right? We reduce retroviral risk to eliminate the antigens in the porcine genome that result in hyperacute or acute rejection. And then we add human genes to promote long-term graft survival. And collectively, right, we think we produce the best, most compatible porcine donor. And these are the donors that are leading to this, 700 days of post-transplant survival in the monkey model.
And then to take it to, like you said, the next step is to just start looking for people that would qualify for this.
I think so. Yeah, so what the FDA has asked for is what we call GLP studies. So there's studies that are done under good laboratory practice. All the studies that we've done to date are more research studies, but we put these transplants in a regulatory environment. This is the data the FDA needs to see. When we complete these GLP transplant studies, that will form the basis for our what we call investigational new drug application, or IND, that we would submit. Right now we're targeting towards the end of 24. One of the challenges in the kidney program, it's almost like a side effect of our success, is because we've had such good success in the primate, the FDA is looking for 12 months of survival data, right?
That came up in the talk, yep.
Yeah, so we have to replicate the data that we've generated so far under GLP. And that, unfortunately, it takes 12 months. So you can't generate 12 months of data in less than 12 months.
No, it doesn't work.
Unless we have a time machine, which we haven't figured that one out yet.
When you go back and look at what patients go through, what kidney patients and kidney failure, and even the ones that have had a transplant, there is I call a creatinine creep. It's over time, as long as you've had your kidney, there's always something going on in the background that's looking to reject it, get rid of it. And that's not probably firm in literature, but I've seen it in a lot of my friends that get out long-term, and I'm talking 20, 25 years, they start having problems, 10, 15 years. You can't keep that kidney forever. It's just not going to happen.
I always think about it as sort of this like smoldering fire, right? Where
exactly,
right, the immunosuppression and how we take care of organ transplant recipients keeps all that kind of under control, but it's always there, right? It's always trying, right?
It's always there.
It's always there. And so, and this is, I know, the team at Tolerus is working on tolerance approaches to try to get rid of that smoldering fire, right? So that it isn't always there. And this is why we think the future of xenotransplantation involves the addition of regulatory transgenes and additional edits to reduce that fire, to reduce the smolder. And, it's interesting, to talk to David Cooper, David Sachs, these are two, surgeons who've been in xenotransplantation for, 40, 50 years.
Right.
Their point is we can keep studying this in monkeys for another 20 years, right? But we won't still know what happens when we go into people, right? And for us as eGenesis with our engineering platform, we have lots of ideas of how to continue to edit the porcine genome to improve compatibility and promote long-term graft survival, but that's in the monkey, right? We need to go to people to understand what are the things that are happening in people that would inform how we need to continue to edit the porcine genome to improve compatibility, to your point, to get rid of the smolder. Because our ultimate goal here is to try to produce organs that reduce the need for immunosuppression, right? And promote long-term graft survival. A great goal, right, would be a 25-year kidney. I mean, that would, if we could reproducibly produce, there's lots of anecdotal data of people, as you mentioned, with long-term success. There are also lots of folks who don't have long-term success.
There are a lot more.
We always put out in the spotlight, the long term, which is great for patients. I mean,
Sure.
it's great for that patient, but unfortunately, as you said, right now, all grafts will eventually fail, right?
Yep.
That's just the way it works.
Yep.
And the goal, and when we think about, the possibility of producing organs to realize this vision of making them as routine as hip and knee replacement, we believe that Xeno actually is the only technology that has the potential to provide that, because we can edit, right? Finding matches, right, of patients, for living donor or decedent donor is challenging, right? I mean, when you find it, it works great, right? I mean, it can be done and the outcomes are beautiful, right? But it's not available for everyone, and likely never will be, right? Because of just the way the matching works, right? And Xeno has the potential, with the right editing tools and understanding what we need to do, to really produce a renewable source of organs that has that compatibility. Now, that won't be the first generation product. I mean, we want the best success possible in this first gen, but again, we've never been in people, so we don't know what are the rejection mechanisms that are going to, be a challenge. We have some ideas, of course, but until you do the experiment, you actually don't know.
You've had focus groups within your corporation or with your program with patients, and you've been very, very careful to involve patients in this whole process every step of the way. And I want that to be known. This isn't just to placate those. You just always have to have patients involved. You do have to, because they're living it, right?
Not only are they living it, they're definitely, absolutely. And as we think about the product, and we think about the first trial, and think about who would want to enroll,
right?
Like what's on their mind? Like, what is a trial that they would want to participate in versus one they would not, right? And what do patients need to see? I mean, we need to see a certain thing, FDA needs to see a certain thing, but the patient who's going to come and sign up for this thing, what do they need to see? Like, because it's their life, they're the ones that are actually going through it. It's very important to have the patient voice in that design of the trial, because I've been involved in studies where we didn't do that, and we show up with a trial that nobody wants to participate in because
no skin in the game, so to speak.
Exactly. And then I think you also have to balance some patients, if that are have failed an allo, they're over 65, they want to go tomorrow, right? So they almost don't care. They're like, whatever it is, I'll do it. We also have to watch that too, because they're willing to do just about anything. And it's up to us to make sure whatever we're doing has the potential to benefit patients. To take that enthusiasm, right, for wanting to help and participate, and really leverage it into something that could be useful.
That's the most important thing I think that eGenesis is doing right now in the patient world, is making a product that getting it right so that it works. All right. This isn't just a run for money. It's a run for life. And I think that's an important thing to remember.
And the challenge is, when you use the primate as the model, you never know, right, how it's going to go in people, right? And we can look at lots of drugs that looked great, before we got into the clinic, and then we got into the clinic and it didn't go so well, right? So I do think we have to, eyes wide open on the risk and potential benefit, but we have the best intentions and things still can go poorly, right? But we're really encouraged by the primate data that we have. And you reach a point where you're not sure what else you could do before you go to a clinical trial to de-risk, right, for patients. I think the other advantage, when you think about the first steps in Xeno, the advantage of kidney transplant is you can go backwards, right? So if the graft doesn't work, right, the graft can be removed and the patient could go back on dialysis or receive an allo transplant. In the case of, for instance, heart transplant, that's not so much, right?
Yeah.
It's harder to go back in a heart transplant.
But in the course of things, it probably is, like you've said, easier to do a porcine heart than it is a porcine kidney. Is that a valid statement?
Well, I would say from an immunological or rejection perspective, they're equally difficult. But from a compassionate use, making a case, where a patient is sort of desperate for something, they're imminently going to die. Yeah, exactly.
Yeah.
And you see this in the heart transplant world more than you see it in kidney transplant. Yeah.
Well, kidneys, they're so vascular. Everything is so really, fine tuned, and it's a world of chaos. But I guess God knows exactly how he wanted it to be, because it's pretty difficult to understand. They've been working on it, in my case, with FSGS for over 50 years, trying to, come up with the reason why it even happens.
Yeah. No, it is. The kidneys are amazing. I mean, absolutely amazing organs. Their ability to concentrate urine and clean. It's really, and this is why we think, when people talk about bioprinting and 3D printing organs, and which is all very interesting, but when you look at the complexity of what a kidney actually is, it's hard to see how that technology is going to be ready anytime soon, relative to xeno. I think, again, xeno, your point about 50 years, xeno's been in the works for hundreds of years, right?
17th century, I read.
Yeah.
I mean,
and all different versions, right?
Yeah.
Right. Yeah. And it's our knowledge now obviously is, way ahead of where it was, but there's still things to learn, right? We still don't know what's going to happen in the first human trials. And, but I think we are closer than ever to actually do that. I mean, we have a path at eGenesis to do one, right? And we now have, guidance from the FDA. We're currently trying to produce our clean donors for that first trial, doing the GLP tox studies. So doing all the bits and pieces that we need to do to run a real trial. Because I think one of the challenges with compassionate use is, one patient is difficult to interpret, right? And so you really want to be able to run a trial that includes several patients, because each patient is so individual, right? And the outcome in one patient may not predict the outcome in another.
Right. So what are you looking for as a block of patients? I know you term age and, longevity on dialysis and stuff like that. What are you looking for the bulk? Are we looking at 5 patients, 10 patients, 20 patients?
Initially? So we had proposed 6 patients to the FDA. The FDA came back and proposed 3 for the first trial, right? And so this is a continuum, right? So when we say first in human, we're looking primarily at safety, right? So if we can do a kidney transplant in a patient with a porcine kidney, what happens from a safety perspective? Once we get through the safety question, then we get into the efficacy question. Now, the challenge in transplant is those two things are very entwined, right? Mm-hmm. Safety and efficacy are kind of one and the same in many ways. We think once we got through the 3 patients, then we would do what we call phase 2 or phase 1B, or basically do more patients, depending on the outcome in those first 3. Right? You can make scenarios where the first 3 go poorly, and maybe we don't go and do more right away, right? Or they go great and we do many more right away. Right? It really depends on the outcome from those first 3. And then the FDA is asking us to put a gap between, right? So it's not like we can do 3 on Monday and then watch all 3 go. In the post-transplant period, we'll do 1, watch that patient closely for 6 months, then do another one, watch closely for 6 months, and then do another one.
By that, you'll have 18 months out.
That's right. The first patient would have 18, the second patient would have a year, exactly.
Mm-hmm. Well,
yeah.
See, and so that's normal, because what people are looking for, not the general public, but what patients are looking for is, there's not a lot of time when you're at end stage. All right.
Yeah.
And not a lot of time. And that's got to be, if you quit looking, you'll never find the answer to it. So you have to start somewhere, and there will be people, I guarantee you, that will want to do this. I think I can tell you without any problem that I would want to, if I was I couldn't really even do an allograft, wasn't in the cards for me, or it was a point that it was available and it was something that research could help with. And that's really where we're at as a patient.
We've heard that from several patients, right? Where again, they've been through allo, or they found themselves on the 65 and they're on the wait list, and they know the statistics, right? They're eyes wide open, right? Sure. And this is an option that may help them, or minimally help somebody else, right? Help the field, help us understand kind of where we need to go as far as engineering. One of the reasons I joined eGenesis is because the monkey transplant model is what we call translatable, right? Pretty translatable. So when you look at what the outcomes in, for instance, allo transplantation, like how do we even get allo? Well, we did it first in animals, right? And the outcomes that we see in the monkey are relatively predictive of what we see now in the clinic. So if we get long-term survival in the monkey, we can anticipate that some patients in the clinic should have long-term survival. Now, we could be wrong and we could miss something, but that gives us a reasonable amount of confidence, about as much confidence as you can have about the potential for this in the first trial. But again, eyes wide open, it may not work at all, right? Or it may work incredibly well. And we just won't know really until we do it.
That's why we're doing the research.
Yeah, no, exactly.
Yeah, there's been a lot of things in medicine that haven't worked, that go by the wayside, but that are rebirthed later on and work. There's ways to,
well, and we will often go back to the early days of allotransplantation, right? 50 years ago, when Starzl and everyone was doing the first transplants, the first few didn't go well.
No, they didn't.
Right. And they had to figure things out. And if you actually look at the data, it didn't take them too long to figure out how to make them better, right? Relative to other fields of medicine. And now, as you talked about before, you can have profound outcomes 20, 25 years post-transplant, which is absolutely amazing considering from where we came, which was, you're either going to spend time on dialysis or it would result in death. I mean, that was the path we were on. That was it, right? And so we think we can actually move maybe a little quicker because we're leveraging all of the years of experience in allo, right? All the immunosuppression work that's been done, right? All the understanding of how to do transplants, how to care for patients in the post-transplant period. Like all of that knowledge that we gained over 50 years, we can leverage in Xeno. And I actually think you saw it in the first heart transplant, right? Where Mr. Bennett survived for 60 days post-transplant, which was longer than the first ever human heart transplant, which was 18 days, right? So that's like one little indicator that, I think Xeno is starting from a better place in many ways than allo did, 50 years ago.
I'm sure that you're well aware of the fact that there's a lot of distractors here from an ethical standpoint. And just in our limited amount of time, maybe that's a subject we ought to talk about later on, but because I'm going to have you back. Anyway, just give me in general, where's the ethics line? Where's the red line here in this research? Is there a red line?
But I think there's always a red line. But I think in Xeno, the use of animals to support human health has been, in our culture for, millennia, right? In addition to, obviously some of us, consume animals, but, look at the early days of insulin, right? The first insulin treatments came from a porcine source, right? And changed the lives of patients with type 1 diabetes. So fundamentally, we look at this as, the use of animal products to support human life, I think is ethically justifiable. I think and I think the use of science like CRISPR to improve outcomes in the use of animal products to support human life is also completely justifiable, right? We're looking for the best outcomes to support human health that we can achieve. Now, we want to do that in a responsible way, right? So one of the things, like, for instance, the FDA is very concerned about, and as are we, is that our edited donors don't make it into the food supply because we just don't know. One, they don't need to, right? Because we can control them. And we just don't know what would happen if we suddenly introduced our animals into the food supply. Now, that said, Revivicor got approval from the FDA for a genetically modified pig that has one of the sugar enzymes inactivated, what we call alpha-gal, and it's the gal-safe pig, right? And it's potentially useful for people who have an allergy to pork, right? And so now they can actually eat pork from this pig. But I think from the ethical perspective, the editing that we're doing, the use of animal products to support human health is all justifiable. We take very good care of our donors. Animal welfare is really important to us, not only from the ethical perspective, but producing quality products and producing quality science, right? That maintaining a very high standard for how we do our science is fundamental to everything that we're doing.
Yep.
So we are comfortable, ethically where we are. We also do, similar to our patient advocacy group, we have a ethics committee, part of eGenesis, some of world leaders in the thinking around xenotransplantation, and we consult that group on everything we do. And as we think about the first trial, and the first patients coming in, informed consent is really important. So patients really know what they're signing up for. because, coming back to the enthusiasm, patients want to help. Patients want to help not only themselves, but also their colleagues, friends, and other patients in the transplant space. We have to be careful that we don't take advantage of that enthusiasm, right? And ask patients to do something that we shouldn't be asking them to do. And this is where I think informed consent, working with institutional review boards, as well as our ethics committee, is really important to us as a company.
That informed consent is more than just a paper that you sign. It's an explanation and a grassroots way of making people alert to the fact that there are just problems with this, or there, could be really dangerous, but it just allows the person to make an informed consent. That's exactly what it is. Make their own decisions.
And there's a lot we don't know, right? And trying to lay out some of the unknowns, right? We want the best outcomes we can get. And as we talked about before, sometimes you don't get them. And just to be eyes wide open of what the potential risks are. And as we design trials, we want to support patient health and mitigate the risk to patients. And then one thing the FDA always is looking for is, okay, what if this goes wrong, right? What are we going to do? What's the back door for patients? What is the remedy, right? If this organ fails during the transplant, right? Let's say they do the transplant and we see instant rejection. What's the plan? Like, what are we going to do? And the FDA wants to see that throughout that post-transplant period too. Like, now things can happen we don't expect, right? But we want to have a forward-looking plan to say, okay, how will we deal with things that don't go as we expect them to go? Because we've all been this long enough, it can happen, and we need to be prepared for it.
Okay, all right. I have things floating in my head and that I want to ask you, but I think getting this type of information going, it's better to have you back later on, or somebody from eGenesis to talk directly about, precise genetic integration. I'm open to that. I really appreciate you, Doctor. It's just fascinating to me because it allows the patient an option. All right. This isn't about eGenesis. This is about the patient. And then that's most important, that you give someone an option. And kidney patients don't have a lot of options. They're getting better, and they're at the forefront now. And I think this xenotransplantation has traction.
No, we agree, and we agree to your point. If it's an option, and it's hopefully an option we can develop over the next couple of years to make it real for patients. I mean, that's the goal.
Anything that you would like to add?
No, we appreciate your interest in what we're doing. And, again, we definitely have patients in mind here, and we want to create products that benefit patients. The science is very cool. I think it's all very cutting edge, but if we're not helping patients, then it's just a science experiment, right?
Yeah, that's exactly right. You hit it right on the head, Mark.
Yeah, and this is way beyond a science experiment.
Yeah. It's way beyond an article in the American Journal of something. That's
right. I think we're close to understanding, how Xeno can help patients. I think we're very close to that. Starting to answer that question.
Well, we'll discuss this later on down the road, you and I, and
sounds good, Ken.
Come up with some ideas. Okay.
Very good.
Listen, folks. I want you to listen to this podcast, and if you are, I want you to share it. And there's no reason to hold back. If you feel like this is of interest to you, get a hold of me. We can put you in touch. If you're currently right now struggling as a kidney patient, if you have anything in your mind that's just unanswered, or you want to just ask the question, give us a call at Kidney Solutions. We're a not-for-profit, and we charge nothing. We don't totally into being funded by the public, and so if you have a question, we can get an answer for you. We have a wealth of information, and we have a wealth of contacts. 830-285-2140, or go to our website, kidneysolutions.org. This podcast will be available pretty soon, real soon, and just friend us and really work with us and spread the word. This is a great thing to be talking about. Okay, so until we come through and talk again, I want you to keep breathing.